Good morning, students. In today's class, I am going to continue the second topic, biological classification. Let us talk about the characters of next class Ascomycetes. Ascomycetes are also called the sac fungi. They are also called the sac fungi. General characters of Ascomycetes. The first character is the plant body of Ascomycetes is unicellular or multicellular. They may be unicellular or multicellular. Example for unicellular organism, yeast. Yeast is the only unicellular fungi. And for multicellular, penicillium. So the members of Ascomycetes are either unicellular or multicellular. Second character. They are saprophytic, decomposers, parasitic, or coprophilous. So then, what is the meaning of coprophilous? Coprophilous means the organism which grow on animal dung. They grow on animal dung are called the coprophilous. So let us move the next character. The mycelium is branched and septate. Mycelium is branched and septate. Let us draw the vegetative structure of Ascomycetes. This is the structure of mycelium with septa. Each compartment has single nucleus. That's why we used to call uninucleate, septate, branched mycelium. This is the vegetative structure of the Ascomycetes member. Next character. Asexual reproduction takes place by the conidia. Conidia are the asexual reproductive spores. And these conidia are produced exogenously on a special mycelium called conidiophore. Conidia are produced exogenously on a special mycelium called conidiophore. Conidiophore. Let us see the structure of the conidiophore with the conidia. There is a vertical growth of a mycelium which is also septate. It may be single or it may be branched. Single or branched. And these branches again develops a small branch-like structures, and these branch-like structures are called the medullae. They are called the medullae. Above this medullae, somewhat same structure, little bit bulged. Structures are there above this medullae, which are more or less look like medullae only, and these structures are called, on the above the medullae there are some bulged structure which are look like medullae only. These are called the stalagmata. And above this stalagmata there is a chain-like structure on each stalagmata. And this chain-like structure is called the conidia. It's called the conidia. When a single conidium detach and fall on the ground, it develops into vegetative mycelium. So these are the asexual spores. Next is sexual spores. So we will discuss about the ascospores. Sexual reproduction results ascospores. Next character is ascospores are spores of sexual reproduction. And these spores are produced in a sac-like structure called ascus. And how these ascospores are forming? Let us discuss in detail by taking an example of yeast. There are three life cycles. One is haplontic life cycle, diplontic life cycle, and haplodiplontic life cycle. The species of yeast show all these three types of life cycle. For example, haplontic life cycle is shown by the yeast species Sachochromyces, and diplontic life cycle is shown by the Saccharomycoids Ludwig, and haplodiplontic life cycle is shown by the Saccharomyces cerevisiae. But to explain the ascospore formation, now I am taking the help of haplontic life cycle expressed in the yeast species Saccharomyces. So original parental cell is haploid. Parental cell is haploid. And this haploid cell undergo a process called the fission. And because of that, what happened? A single cell divides into two cells. There are two cells are forming, both are haploid. By the process called fission, a single haploid cell undergo fission and forms two haploid cells. And these two haploid cells fuse together. And that process is called the hologamy. Because two haploid cells are fusing. These are not gametes, these are complete cells. Because these are originally haploid, so there is no need of meiosis. And these two haploid cells are fusing. That's why this process is called the hologamy. And fusion results in a diploid cell, and that we call the zygote, which is diploid. And this zygote only called as ascus. This is called the ascus. And in diploid zygote, meiosis takes place. Meiosis takes place and that results in 4 cells. 4 cells which are surrounded with the cytoplasm. And these cells are called the ascospores. But sometimes what happens? There is a formation of 4 more ascospores is also there. Means by meiosis in zygote 4 ascospores are producing. But sometimes there are 8 ascospores are also there. And how these 8 ascospores are forming? Soon after this meiosis, these 4 ascospores are next entering mitotic division. And because of mitotic division following meiosis, there are 8 ascospores are forming. Already 4 are there. Because of mitotic division, 8 ascospores are forming. 8 ascospores are forming, which are surrounded with cytoplasm. When these ascospores liberate, they form the parental cell. So this we call the haplontic life cycle because the maximum stages of this life cycle are in haploid stage except this zygote. The remaining all stages are in haploid condition. So that's why this life cycle is called the haplontic life cycle. So ascospores may be 4 if there is only meiosis. There are 8 if meiosis is followed by mitotic division. Means there may be 4 or 8 ascospores. If only meiosis, 4 ascospores. If the meiosis is followed by mitotic division, then 8 ascospores are formed. So in this manner, ascospore formation takes place. And these ascospores are present in ascus, and ascus are producing in different types of fruiting bodies, and those fruiting bodies are called the ascar. They are called the ascocar. Example, aspergillus, claviceps, and neurospora. These are the different types of, for example, for the Ascomycetes group. So neurospora is the one which is extensively used in biochemical and genetic work. And the last character of this group is there are some members like morels and trophels which are edible. These are edible and are considered as delicacies. Means they are very costliest edible food. So these are the characters of the class Ascomycetes. Let us differentiate Ascomycetes and the class Phycomycetes. So we can make out what are the characters which are similar or dissimilar in these two classes. Let us see the difference between these two classes. The first difference between these two, or may say the common name of these two. Phycomycetes is commonly called algal fungi. Algal fungi. Whereas Ascomycetes are called the sac fungi. They are called the sac fungi. Next character. So mycelium is aseptate, cenocytic or multinucleate. Whereas in case of Ascomycetes, mycelium is septate, uninucleate. Septate and uninucleate. And third character, asexual spores are zoospores and aplanospores. Here asexual spores are conidia. Asexual spores are conidia. So these asexual spores are produced endogenously. But conidia are produced exogenously. They are produced exogenously. Next is sexual spores are zygospores. Whereas sexual spores are ascospores. So these are some of the characters which differentiate class Phycomycetes and class Ascomycetes. So here I am concluding this topic. So in the next class we will continue the next class Basidiomycetes. Thank you.